I want to present two examples of applications where we have been using the visual acute method and with pretty good success. The first one is a project that we are taking in the Rare Fantasy Center about a preclinical study for gene replacement therapy in a mouse model for multiple sulfatase deficiency. So first of all, what is a multiple sulfatase deficiency? So this is a rare disorder. It's a lysosomal storage disorder. It mainly affects the brain, skin, and skeleton from usually kids. Is caused by the loss of function mutations in one single gene, the gene called SAMF1. It's an autosomal and recessive inheritance. So it's considered an ultra-rare disease. By now, we have only about 50 living cases identified worldwide. And the disease varies, the phenotype of the disease varies a lot depending of the kind of mutation in the SAMF1 gene, where you can have cases with neonatal onset, late infantile, and juvenile. In general, it involves loss of mental and motor abilities, and then also is characterized by a progressive vision loss. And by now, there is no cure available. So the gene SAMF1 is a really key gene in the metabolic pathway for the activation of sulfatase. So if the key gene in charge of the activation of 17 downstream sulfatases, that's why it's so severe, the phenotype caused by deficiencies in SAMF1. In terms of the mouse model that we have, back in 2007, a team led by Dr. September et al. Developed a new mouse knockout for the SAMF1 gene. And this knockout mouse was able to represent several phenotypes seen in patients. However, the model in general is far more severe than we can usually see in most of the patients. The life expectancy of the mice, this lifespan is about 30 days in average, but we can always find some mice that can escape and have longer survival. The mice also show craniofacial features and a reduced body size. For our study, what we choose to do was starting with this knockout SAMF1 mice. We decided to test the ability of a new viral vector, AAV9 SAMF1 construct that should be able to replace the deficient mouse SAMF1 gene for the human SAMF1 version. So we did injections in the mice by intrathecal administration at postnatal day 7. We also tested the potency of the virus at the same time. First, so comparing the highest dose with undiluted construct to 3 different dilutions of the same viral vector. So we performed a continued monitoring of survival, growth, and some behavioral outcomes during the lifespan of the mice. And we have an endpoint fixed at about 18 months of age. So this is kind of partial preliminary data that we have. The mice is almost reaching to the endpoint. So far what we have is that the survival in mice injected with undiluted viral vector, or even the first dilution of 1 to 4, was able to rescue or improve the survival and lifespans of the mice by having a 60%, about 60% survival of more than a year after the injections. We also saw those response event result regarding to survival. So it's clearly a dose response dependence for survival in these mice. So what the most important part that I would like to share today is what we actually found in these mice. So as I said before, in humans, it's very common that patients develop a progressive vision loss. So we also noticed this vision loss in the knockout mice. As you see in this graph, 9 months of age, knockout mice untreated shows a huge, significant reduction of the visual acuity threshold compared to wild types and untreated. But if we look at the knockouts treated with the highest dose of AAV9 via intrathecal, or even treated with a low dose of AAV9, the mice are able now to have a more a better vision threshold compared to wild types, and it's totally comparable to wild types and treated controls. So, however, when we look at these mice again 3 months later, we notice now that actually the mice that received the low dose of AAV9, they have a poor, very low threshold. So the benefit of the rescue that we were able to have was totally dose dependent. And probably the most important fact is that the high dose injected of AAV9 was able to almost 100% rescue the vision phenotype, as you can see, compared to the wild type controls. Next is a cardiac phenotype that we also find in these mice. This is thanks to the help of Jack in the CBA. He was able to run electrocardiograms in these mice. We found that actually the knockouts mice show a low heart rate. As you can see in this graph at 9 months to the left. But when we injected the mice with the highest dose intrathecal, we actually were able to rescue this phenotype, and now the mice has a better cardiac function. However, when we look at the 12 months time point here to the right, the best rescue was achieved with the highest dose of AAV9. And once again, we see that the benefit is totally dependent of the dose injected. So to summarize this section, we found how the AAV9 SAMF1 gene therapy can improve survival, extend lifespan, and by now we don't see any signs of neuropathy. We also see that the gene therapy approach is effective, targeting the CNS, and it shows we are able to find improvements in ophthalmic and cardiac phenotypes. We also noticed that how the benefit in these two phenotypes, it was totally dose dependent. and how the best benefit was achieved with the highest dose. And by now, we haven't seen any signs of toxicity or cytotoxicity in the mice after more than a year of treatment. And we strongly consider that this will be will be predicting a good potential benefit for pediatric subjects and future clinical study designs.